So this talk is going to look at cellular structure and function as it pertains to common clinical situations that you may encounter in the emergency department. And at the end of the lecture, you'll be able to describe the main functions of the human cell, the key structures that make up that cell, and the impact of various clinical scenarios on the human cell. So let's think about the functions of the human cell. So if you wish, you can pause the video at this stage and just try to make a list for yourself before we proceed. Okay, let's see what we have here. So the cell provides a structure in which the metabolic functions of the body take place. Cells, both individually and as part of organs, can take in nutrients and then utilizing those nutrients produce energy. They also have specialized functions, some of which we'll talk about, but then, if you think about all the different organ systems in the body, each is specialized towards a certain function that the body requires. Most cells have the ability to replicate, and of course they encode the genetic material of the organism. So this is a picture of an animal cell showing some of the more important organelles, and we're going to focus today on the nucleus, the smooth and rough endoplasmic reticulum, the mitochondria, and then a little bit about the Golgi apparatus as well, and of course the cell membrane. So the cell membrane is a bilaminar phospholipid layer with a lipophilic hydrophobic outer layer and a hydrophilic inner layer. And this is really important in maintaining cellular integrity. So it acts as a barrier to chemicals and molecules outside of the cell membrane, and also as a selective transport medium for chemicals and molecules. And actually, you need to know a little bit about membrane transport and the different types of membrane transport, comparing passive transport, which includes passive diffusion, osmosis, and facilitated diffusion, active transport, and endocytosis. And you will need to read this up and have a general idea about which types of molecules are transported in which way. So as we go through this lecture, we're going to find a series of questions. I would like you to pause the video, attempt the question, and at the end of the lecture we will go through the answers. Here are your first two questions. Pause the video. You have 1.5 minutes for each question. Alright, let's talk about the mitochondria now. So the mitochondria are small organelles inside of the cell which actually function quite separately from the rest of the cell. They're not produced by the cell nucleus, they're not encoded in the cell nucleus, and they actually sort evolutionary wise to be small organisms that may have invaded cells and set up a symbiotic relationship with the cells. So what are the functions of the mitochondria? So mitochondria are key in energy production, and the mitochondrial inner membrane is the site of the Krebs cycle, aerobic respiration, and the electron transfer chain. So they're really important in producing ATP due to aerobic respiration. They're also important in maintaining calcium homeostasis. They play a part in innate immunity, and similarly, they may be important in programmed cell death and in the process of cell differentiation. Again, I'd like you to pause the video, and for each question you have 1.5 minutes. Attempt an answer, and we'll review this at the end of the lecture. Okay, so ribosomes and the rough endoplasmic reticulum are key to the production of proteins. So this is where the DNA from the nucleus is the code is brought from the nucleus to the endoplasmic reticulum by messenger RNA, and then transfer RNA uses this code to synthesize protein by assembling individual amino acids into chains. Once this happens, the chains then fold on themselves to form tertiary and quaternary structures, and then which form what we know as complex proteins. These proteins are then transported to their sites of function or to the cell membranes for uses external to the cell by, in part, the rough endoplasmic reticulum. The Golgi apparatus and the smooth endoplasmic reticulum are also important in the creation of complex molecules, particularly lipids, phospholipids, and lipoproteins. The Golgi apparatus, the smooth endoplasmic reticulum is also important in the production of glucose from glycogen and in calcium metabolism. The Golgi apparatus also plays a part in lipid production and in packaging of complex molecules such as proteins, lipoproteins, phospholipids, so that they can then be exported from the cell. So this is the general layout of protein production. The code is stored in DNA within the nucleus. It's then transcribed to RNA, which is then used to assemble amino acids into protein. And this on the right is just a more detailed representation of the same process. And remember, you have messenger RNA taking the code from the DNA in the nucleus into the cytoplasm, and then transfer RNA helping to assemble the amino acids in the ribosome, and then post-translation modification to create the more complex structures of these chains of amino acids to produce proteins. When we talk about protein structure, we talk about primary, secondary, tertiary, and quaternary structure. and it's just about the spatial orientation of the amino acid chain. So the primary structure is just the sequence of chains of the amino acid, whereas the secondary structure is the initial folding of the amino acid chains into simple structures such as an alpha helix or a beta pleated sheet. And then tertiary structure is the three-dimensional configuration of this protein chain, whereas the quaternary structure is the conglomeration of more than one protein chain. And if you look through this again and you try to apply it to the structure of hemoglobin, you will see what is meant by each of these structures. Questions on protein production? Pause the video, a minute and a half for each question. You'll see the answers at the end. Finally, let's think a little bit about the nucleus. So it stores the genetic code and in that way regulates cell structure and growth. You need to know the phases of mitosis from interphase through telophase, and you also need to know what happens at each part of the phases of mitosis. Two questions on the nucleus. Again, pause the video and attempt these questions. Just a little bit about aging. So aging is a planned process of the cell, and what you see here are different built-in mechanisms to produce cellular aging and programmed cell death. A little bit about cell senescence. It is a way of limiting the lifespan of a cell due to arrest of the cell cycle, which is a preventive strategy in aging process. As cells continue to age, they are more likely to develop abnormalities or nuclear and structural changes which may lead to disease such as cancer. There are multiple stimuli that produce senescence, and they're maintained by certain proteins which are in the tumor suppressant pathway. This process suppresses malignancy and of course increases with age. Two questions on cell aging. Pause the video and have a go. So if you have any questions, you can bring them up in class. Let's go. So question 1. What's the function of glucose in oral rehydration salts? So the answer is it enhances absorption of sodium by an active co-transport mechanism. Question 2. Using local anesthetics, the pH in alkaline tissue is closer to the pKa of the drug and allows more bioavailability at the cellular level. What about the mitochondria? Well, ethanol overdose does not cause mitochondrial dysfunction, whereas the others do. And one of the symptoms least likely in a patient with aspirin overdose is metabolic alkalosis. The primary problem with sickle cell disease is at the primary protein structure level. In rhabdomyolysis, alkalization stabilizes myoglobin. Question 7, the answer is C. Question 8, the answer is A. Question 9, the answer is B. And question 10, the answer is A. You can go through this later on just to double check your answers. So in conclusion, we talked about cellular structure and function and the process of cell aging. Thank you.